Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain PU Board Biology Model Question Paper for Karnataka State, and it is released by PU Board based on reduced syllabus for only 2020 and 21 batch. According to reduced syllabus, the weightage of each and every chapter is: the first chapter, Reproduction in Organisms, for 2 marks. Second chapter, Sexual Reproduction in Flowering Plants, it is for 13 marks. Third chapter, Human Reproduction, for 11 marks. Fourth chapter, Reproductive Health, for 6 marks. Fifth chapter, Principles of Inheritance and Variation, for 14 marks. Sixth chapter, Molecular Basis of Inheritance, for 14 marks. Eighth chapter, Human Health and Diseases, for 12 marks. 10th chapter, Microbes in Human Welfare, for 7 marks. 11th chapter, Biotechnology, Principles and Process, for 9 marks. 12th chapter, Biotechnology and its Application, for 6 marks. 13th chapter, Organism and Population, for 8 marks. And 15th chapter, Biodiversity and its Conservation, for 4 marks. The question paper is given for 105 marks, but the students have to score 70 marks. In this, part A, Roman number 1, answer the following questions in one word or one sentence each. Totally 10 questions are there. Each question carries 1 mark, totally 10 marks. Question number 1. What is the function of tapetum? The answer is, the innermost wall layer in anther wall is called tapetum, and it nourishes the developing pollen grains. Question number 2. Define spermiation. The answer is, after spermiogenesis, the sperm head it become embedded in the Sertoli cells and are finally released from semiferous tubules by a process is called spermiation. Question number 3. MTPs are legally restricted in our country. Justify by giving reason. The answer is, due to emotional, ethical, religious and social issues involved in the it, Government of India legalized MTP in 1971 with some strict conditions to avoid its misuse. Question number 4. Name any one autosomal recessive disorder. The answer is sickle cell anemia. Sickle cell anemia is a autosome linked recessive disorder. Question number 5. Which RNA is also called adapter molecule? The answer is, the tRNA molecule is called an adapter molecule because that would on one hand it read genetic code and on another hand it brings a suitable amino acid for protein synthesis. Question number 6. Cancer patients are treated with alpha interference. Give reason. The answer is, the patients are given with substances called biological response modifiers such as alpha interference, which activates their immune system and helps in destroying the tumors. Question number 7. Name the pathogenic virus which is used as a biocontrol agent. The answer is, the majority of baculoviruses are used as a biological control agents or in the genus nucleopolyhydroviruses. Question number 8. What is RNAi? It is RNA interference. The answer is, RNAi is a method involving in silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents the translation of the mRNA is called RNA interference. Question number 9. Write the equation for representing logistic growth. The answer is d capital N by dt is equal to R capital N bracket K minus capital N by K. It is the formula for logistic growth. Question number 10. State the result of David Tillman's long-term ecosystem experiment. The answer is Tillman found that plots with more species showed less year-to-year variation in total biomass, and he also showed that in his experiment increased diversity contributed to higher productivity. It is the result of David Tillman's with a long-term ecosystem experiment. So these are the 10 1-mark questions from part A. Part B, Roman number 2, answer any 5 of the following questions with 3 to 5 sentences each wherever applicable. 5 into 2, totally 10 marks. Question number 11. Differentiate perisperm and pericarp. The answer for this question is the first difference. The residual persistent nucellus is called perisperm, but the wall of the ovary it develops into wall of the fruit is called pericarp. The second difference is some of the seeds such as black pepper and beet ruminant of nucellus is also persistent is called perisperm, but in pericarp it may be fleshy in guava and dry in groundnut. These are the two main differences in between perisperm and pericarp. Question number 2. List the hormones produced in women only during pregnancy. The hormones are human chronicle gonadotropin hormone, human placental lactogen hormone, estrogen, progestogen, and relaxin hormones. These are the hormones released during pregnancy. Question number 13. What is hibernation? Give example. The answer is hibernation is a process by which the animals like bear avoid the stress and become inactive during winter. The bear going into hibernation during winter season. Question number 14. Write one advantage and one disadvantage of external fertilization. The answer is fusion of both type of gametes it occur outside of the body of organisms is called external fertilization. In this, the advantage is large number of offsprings are produced, and the disadvantage is that of the offsprings are extremely vulnerable to predators, threatening their survival up to adulthood. These are the advantages and disadvantages of external fertilization. Question number 15. Write a note on the pollination mechanism in Vallisneria. The answer is in Vallisneria, the female flower it reaches the surface of the water with the help of a long stalk, and the male flowers are pollen grains. They release on the surface of water, and these are carried out passively by water current. Some of them eventually reach the female flowers for pollination. So it is the mechanism of pollination in Vallisneria. Question number 16. Write the scientific name of organism causing pneumonia and ringworm. The answer is pneumonia. It is caused by Streptococcus pneumoniae and Haemophilus influenzae. The ringworm is caused by a fungi. It belongs to the genera Microsporum and Trichophyton and Epidermophyton. Question number 17. Name any two bacteria used as a biofertilizers. The answer is Rhizobium, Azospirillum, and Azotobacter. These are the bacteria used as a biofertilizers. Question number 18. Mention the reasons for infertility. The answer is the reasons for infertility is due to physical, congenital diseases, drugs, immunological, or even psychological reasons.
Part C, Roman number 3. Answer any 5 of the following questions in 40 to 80 words each wherever applicable. 5 into 3, 15 marks. Question number 19 with a split form. First question in 19. Compare geitonogamy and xenogamy for 2 marks. So geitonogamy is a method of transfer of pollen grains occur from anther to stigma of another flower but present in same plant. But in xenogamy, transfer of pollen grains occur from anther to stigma of different plants. Geitonogamy is a functionally cross-pollination but genetically autogamy. But xenogamy, it is a functionally and genetically cross-pollination. Second question in 19. What are Chasmogamous flowers for 1 mark? The flowers which open or expose their anther stigma during maturation are called Chasmogamous flowers. Question number 20. Mention the different blood groups in humans and their possible genotypes having small I allele. In human blood group, by taking genotype of organism and the blood type of organism. If the genotype having capital IA, small I, it express A blood group. The genotype with the capital IB, small I, it express B blood group. The genotype with small I, it express O blood group. So only these are the 3 blood groups possible with the small I allele. Question number 21. List out the accessory glands associated with male reproductive system. The male accessory glands, it includes paired seminal vesicles, a prostate and paired bulbourethral glands. Question number 22. To a user, the contraceptives must be ideal in all aspects. Justify the statement by mentioning the quality of an ideal contraceptive. The answer is an ideal contraceptive should be user-friendly, easily available, effective and reversible with no or least side effects. And also, it should be no way interfere with the sexual drive, desire and or the sexual act of the user. These are the features of ideal contraceptives. Question number 23. So differentiate active immunity from passive immunity. Active immunity is when a host is exposed to antigen, which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body due to active immunity. But in passive immunity, when ready-made antibodies are directly given to protect the body against foreign agents, is called passive immunity. Active immunity is a slow process, but a passive immunity is a fast process, and active immunity takes time to give its full effective response. But some antibodies from their mother body baby it receive through placenta during pregnancy is example for passive immunity. Question number 24 with split form. First question. Differentiate between micro-injection and biolistic method. So micro-injection and biolistic methods are used in biotechnology to transfer RNA molecule into host cell. In case of micro-injection, the recombinant DNA it is directly injected into nucleus. But in biolistic method, cells are bombarded with high velocity microparticles of gold or tungsten it is coated with or DNA molecule. And second difference is this micro injection method is suitable for animal cell, but a biolistic method it is suitable for plant cell to transfer or DNA molecule. And second question in 24 for 1 mark. Give an example for disarmed pathogen vector. In disarmed pathogen vector, which can allow the infect the cell, transfer recombinant DNA into host. Example, retroviruses. Question number 25. What is biodiversity? Mention the types of biodiversity. The totality of all organisms or a collection of variety of all life forms is called biodiversity. It is three types. One is ecological diversity, species diversity, and genetic diversity. Question number 26. Mention the three reasons of transcriptional unit in DNA. A transcriptional unit in DNA is defined primarily with three reasons in DNA. In this, the first reason is a promoter, second reason the structural gene, and third reason a terminator. These are the three more questions in this model paper. Part D, section 1. Roman number 4. Answer any 4 of the following questions on 200 to 250 words each wherever applicable. 4 into 5, 20 marks. Question number 27. Describe the structure of an anatropous ovule with the help of a neat labeled diagram. In this, answer for this question is the ovule is a small structure attaches to placenta with the help of a stalk. It is called funicle. So the body of the ovule fused with funicle in a region called hilum, and it represents the junction in between ovule and funicle. So ovule generally it is surrounded by one or two protective envelope layers called integuments. These integuments are present around ovule body. It is a stalk of ovule. So integuments encircles the nucellus tissue except at a tip with a small opening. It is called microfile. Opposite to this microfile, it is the end called chalaza. It represents the basal part of ovule. In ovule, a mass of cells are present called nucellus having abundant reserve food materials. An ovule generally has a single embryo sac or female gametophyte after its maturation. So these are the valid points for answer. Along with this answer, you should draw the diagram by mentioning all this labeling like chalazal pole, embryo sac, nucellus, integuments, inner integument and outer integument, microfile, microfilal pole, funicle and hilum. It is the answer for this question. Question number 28. Sketch and label a sectional view of female reproductive system. So it is the diagram you have to draw for 5 marks. In this, only labeling carries half mark, and you should mention more than 10 labelings. It represents ovary, fimbriae, isthmus, ampulla, infundulum as a fallopian tube, cervix, cervical canal, vagina, endometrium, myometrium, perimetrium, and uterine cavity, uterine fundus. So these are the labelings you should mention at least more than 10 labelings. Each labeling carries half mark. Totally half into 10, it is equal to 5 marks. Question number 29. Describe Hershey and Chase experiment. So answer for this question is Hershey and Chase worked to discover whether the protein or DNA from viruses that entered into bacteria during infection. Hershey and Chase, they grow some viruses on a medium that contained radioactive phosphorus and some other bacteria, other viruses on a medium that contain radioactive sulfur. The virus is grown in the presence of radioactive phosphorus. It consists of radioactive DNA and normal protein coat. But viruses which are grown in radioactive sulfur, it contain radioactive protein but normal DNA. So radioactive phases are allowed to attack Escherichia coli bacteria. So during this attack, you should draw this diagram. In this, it is the first virus consists of radio labeled protein capsule, and it is the second virus consists of radioactive DNA. So after infection, blending and centrifugation, radioactive sulfur is detected in supernatant, and radioactive phosphorus detected in cell. It indicates the DNA only enters into host cell during infection. So the viral coat, it is removed from bacteria by agitating them in a blender. Then the virus particles were separated from bacteria by spinning them in a centrifugation. And finally, the bacteria which was infected with virus that had radioactive DNA were radioactive. It indicates that DNA is the material that passed from viruses to the bacteria. It is the answer for this question. Along with answer, you should draw the diagram. So DNA, therefore, the genetic material, it is passed from virus to bacteria. Question number 30. For hereditary diseases, gene therapy is considered as the corrective therapy and justified by explaining gene therapy for adenosine deaminase ADA deficiency. So answer for this question is gene therapy is a collection of methods that can allow correction of gene defect that has been diagnosed in a child or embryo. The first clinical gene therapy was given in 1990 year to a 4 years of girl. She suffered with adenosine deaminase deficiency. So the enzyme adenosine deaminase is crucial for immune system to function. The disorder is caused due to deletion of a gene for adenosine deaminase enzyme in some children. The ADA deficiency can be cured by bone marrow transplantation and enzyme replacement therapy, in which functional ADA is given to patients by injection. But a first step towards gene therapy in this, lymphocytes first collected from blood of patients, and then these lymphocytes are grown in a culture outside of the body. At this time, a functional ADA cDNA with the help of retroviral vector, it is introduced in lymphocyte, then which are subsequently again returned into patient's body. But lymphocytes are not immortal because of short lifespan, so that the patients required the periodic infusion of genetically engineered lymphocytes. So finally, the gene isolated from marrow cells produce ADA enzyme. It was introduced into the cells at early embryonic stage. It could be a permanent cure for ADA deficiency. These are the valid points for this question. Question number 31. Mention the causes and features of Down syndrome. So Down syndrome is caused due to presence of an extra copy of chromosome number 21, and it is example for trisomy of 21. So the disorder, it was first described by Langdon Down in 1866. The sum of the features of Down syndrome is the short statured with a small round head, flat back head and broad flat face. The baby having big wrinkled and furrowed tongue and partially open mouth. The palm is broad with characteristic palm creases and many loops on fingertip. The baby suffered with congenital heart diseases along with physical, psychomotor and mental development is retarded. These are the features of Down syndrome. And question number 32. The population interaction involved in the following examples. Each one carries one mark. The first one is animal eating on plant. It is example for predation. Visiting flaminios and resident fishes of a lake. It's example for competition. And human liver fluke on and snail is example for parasitism. And orchids growing on mango tree is example for commensalism. And flower and its pollinating insects example for mutualism. So each one carries one mark. Section 2, Roman number 5. Answer any three of the following questions in 200 to 250 words each wherever applicable. 3 into 5 is equal to 15 marks. Question number 33. Explain incomplete dominance with suitable example. The answer is incomplete dominance is a phenomenon where both alleles of a characters are expressed incompletely, producing a new or intermediate or blending phenotype in heterozygous condition is called incomplete or partial dominance or blending inheritance. Best example for incomplete dominance is flower color in dog flower plant is also called snapdragon or Antirrhinum majors. In snapdragon, the flower color red, it is expressed with capital R allele. It is a dominant over white with small r alleles. In a cross between red flowered plant with white color plants, the F1 hybrid produce pink color due to heterozygous condition like capital R and small r. A cross between two F1 hybrids, it gives red, pink and white color flowers with 1:1 as a phenotypic and genotypic ratio. In incomplete dominance, the phenotypic ratio, it is equal to red, pink, white. It becomes into 1:2:1. Genotype ratio capital R, small r, it is equal to 1:2:1. Along with this, you should draw this flow chart. It representing phenotype red, white color flowers, genotype capital R, small r, gametes from first plant only capital R, second plant only small r. In F1 generation, capital R, small r, it gives all pink, and the gametes from F1 hybrid having two types capital R and small r, two types of gametes. These are fused randomly to form F2 generation. First combination is capital R red, capital R small r pink, capital R small r pink, small r white. Now we should mention both phenotypic ratio and genotypic ratio like this flow chart. Question number 34. In RDNA technology, unless the desired DNA is separated and isolated, it cannot be introduced in a vector. How separation is achieved using gel electrophoresis? This question carries diagram. In this, the cutting of DNA by restriction endonuclease, it results in fragments of DNA. And these fragments can be separated by a technique known as gel electrophoresis. In this gel electrophoresis, DNA fragments generally having negatively charged, and these negatively charged molecule, they can be separated by forcing them to move towards anode under an electric field through a medium or matrix. In gel electrophoresis, the commonly used matrix is agarose, which is a natural polymer extracted from seaweeds. It is a diagram you should draw for answer. In this, you should mention wells, largest and smallest DNA bands. The DNA fragments separated according to their size through sieving effect provided by the gel piece. The smaller fragments having size, they are further it moves. The separated DNA fragments can be visible with orange color bands after staining with ethidium bromide and followed by exposing to UV radiation. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece is process called elution. So these are the valid points for this question as a answer. Question number 35. Explain the stages of life cycle of Plasmodium. So Plasmodium generally it enters into human body in the form of sporozoites. It is called infectious form through bite of a infected female Anopheles mosquito. The parasite initially multiply within the liver and then attack the red blood cells called RBC, resulting in their rupturing. The ruptured RBC is associated with releasing of a toxic substance is called hemozoins, and these hemozoins responsible for chill and high fever requiring every 3 to 4 days. The female Anopheles mosquito bites an infected person. This parasite again enters into mosquito's body and undergo further development. So the parasite multiply within them to form sporozoites that are stored in their liver or in their salivary glands. When these mosquitoes bites a human, the sporozoites are again introduced into person, thereby initiating the same events. These are the various events in life cycle of this. So malaria parasite generally it need two host organisms like human and mosquitoes to complete its life cycle. So these are the valid points for this life cycle. Question number 36. Describe the structure of nucleosome with the help of a neat label diagram. So the negatively charged double-stranded DNA, it is wrapped around the positively charged histone atomer to form a structure called nucleosome. A set of positively charged proteins with a basic nature called histone. These proteins acquire positive charge. So depending upon the abundance of amino acid residues with the charged side chains, it gets positive charge. These histone proteins are generally rich with some basic amino acid residues like lysine and arginine. The histones are organized into unit is with the 8 molecules is called histone atomer. It consists of 8 histone proteins. So it is a diagram for this question, and it is a histone atomer and a double-stranded DNA. It is wrapped around this histone atomer in 2 turns, and H1 protein binds outside. A typical nucleosome, it consists of 200 base pairs of DNA helix, and the nucleosome constitute the repeating units of a structure in nucleus called chromatin. The packaging of chromatin it need at a higher level, it need additional set of proteins collectively called non-histone chromosomal proteins NHC. In chromatin, some of the regions are loosely packed and stained lightly referred as euchromatin, and some of the regions are densely packed with stained dark called heterochromatin. So these are the valid points for nucleosome structure. Question number 37. Mention the source and function of each for the following. One is penicillin, second one is streptokinase, third one is cyclosporin A, fourth one is statins, and fifth one is lactic acid. Penicillin is isolated from Penicillium notatum. It is useful for potential as an effective antibiotic. Streptokinase produced by a bacteria Streptococcus and modified by genetic engineering and is used as a clot buster for removing clots from blood vessels of patients. Cyclosporin A, it is produced by a fungi, it is called Trichoderma polysporum and it is used as a immunosuppressive agents in organ transplanted patients. Statins produced by yeast called Monascus purpurans have been commercialized as blood cholesterol lowering agents. Lactic acid produced from a bacterium Lactobacillus is used as a chemical in laboratory. So these are the functions of different molecules. So it is the first model question paper released by Karnataka PUC board for students. The questions with suitable answer. Thanks for watching.